Tuisyenonline HR. From my previous video, I already talked about the gene technology. An example is to produce the insulin hormone. In this video, I want to talk about the biotechnology. What is biotechnology? Biotechnology is application of organism, biological system, and biological process in manufacturing and services in industry. Do you know how this garlic become the garlic tablet? And how you know the insulin hormone is produced? And how we get this beverage? Okay, all of that, they are use biotechnology in their industry. Okay, so there are more application of biotechnology in our life. For example, the insulin, we use genetic engineering. Another application in biotechnology in our agriculture, in medicine, okay, to produce the COVID vaccine and so on. Forensic health, DNA fingerprinting, to know the criminal or so on. In food production, in food and beverage, and other biotechnology that we have known in these years. Okay, in this video, I want to discuss about the application of biotechnology in public health and forensics. Okay, let's see my video. Okay, apart from producing beneficial transgenic organisms, DNA technology is also applied in the medical procedure, okay, such as genetic screening. Genetic screening can be defined as the detection of mutant gene in an individual. Okay, one of the situations where genetic screening is particularly relevant to the prenatal. Okay, using genetic screening technique, it's possible to detect and identify certain serious genetic disease in the unborn baby. If such a disease is detected, the parent is usually given a counseling. Okay, the parents are usually advised to choose between two methods of genetic screening. Okay, either amniocentesis or chorionic villus sampling. Okay, for amniocentesis, we take a sample from amniotic fluid, while chorionic villus sampling, we take the fragment of chorionic villus. The test is performed with an ultrasound-guided transabdominal needle that withdraws a sample of amniotic fluid which contains some living fetal cell. So the cells are cultured and grown for genetic analysis. Okay, so the cell culture is more than 1 month. Chromosome analysis can be performed on the culture fetal tissue samples, and DNA can be extracted from them for molecular testing to identify any genetic or chromosomal disease of the fetus. Okay, so you see the set of chromosome. It shows a boy because of chromosome X and Y, and a normal. Okay, normal male. Okay, other method is chorionic virus sampling. The test is performed with an ultrasound guided transvehicular catheter or transabdominal needle that is passed into the developing placenta. Fragments of the chorionic virus are withdrawn through a shrink. Okay, so you see the transabdominal needle is passed through the pelvic to take the fragment of chorionic virus. Okay, and the chorionic virus will withdrawn through shrink. So the cell culture and chromogenetic analysis. Chromosome analysis can be performed on the tissue sample, and DNA can be extracted from them for molecular testing to identify any genetic or chromosomal disease of the fetus. Okay, so you see the set of male chromosome with XY normal. Okay, so the CVS test can be performed earlier in the pregnancy 10 to 11 weeks, then amniocentesis 16 to 18 weeks, and it was only a few days to obtain a result. Okay, so however, the CVS is poses a slightly higher risk of miscarriage and does not offer as much information as amniocentesis. Okay, so by this method we see chorionic virus sampling is more higher risk because the earlier month of pregnancy. Okay, other application of biotechnology is gene therapy. Okay, so what is gene therapy? Gene therapy is a promising technique in treating diseases that are caused by a defective gene. Introduction of functioning gene into this patient to correct their disorder is called gene therapy. The first attempt at human gene therapy was carried out on an infant who suffered the SCID disease. Okay, which called severe combined immunodeficiency, SCID. SCID is the result of the child being homozygous for defective gene encoding in the enzyme adeninosin diamase. Okay, ada the normal catabolism of purine is deficient, and this is particularly any toxic for T cell and B cell. Okay, so ada will cause the abnormal purines, then it's particularly toxic for T cell and B cell. Okay, T cell and B cell. In the procedure, matured T cell were removed from the patient and grown in laboratory. Okay, so this cell were then infected with harmless genetically engineered virus carrying a normal gene for the enzyme adenosine deaminase. Okay, so we take the cell that are defective gene. Okay, and then the engineered virus will carry a normal gene for the enzyme adenosine deaminase. Okay, so the process is doing in laboratory. Okay, so the virus then incorporate their DNA into the DNA of patient cell. Okay, providing a working copy of the gene results. Okay, this genetically corrected. Okay, so the T cell will then return to the patient to produce a therapeutic response. Okay, so virus cell membawa yang normal cell, so dia akan incomplete with DNA patient cell. So they will treat the DNA patient cell that have a disease. Okay, so the patient cell will normal. Okay, so recombinant DNA is the basis of gene therapy, in which genes within cells are removed, replaced, or altered to produce new protein that change the function of the cell. Okay, so the patient is now get a new normal cell. Okay, so the use of gene therapy also be approved in diseases such as cystic fibrosis, muscular dystrophy, and other some cancer. Okay. Application of it is in a criminal case of forensics. Okay, so the process of DNA fingerprinting to solve the criminal case or other issues. As a forensic scientist, you use DNA fingerprinting to compose the DNA from the biological sample obtained at the crime scene. Okay, so what are the biological samples? Okay, so the victim's blood sample, okay, containing rapist DNA or suspect or DNA sample such as blood. Okay, so having collected the necessary sample from the crime scene, okay, so the victim blood, the vaginal sweat, the suspect 1 and suspect 2 blood. Okay, so now is proceed to the lab for analysis. Okay, so DNA is first extracted from the sample. The restriction enzyme is added to the samples to cut the DNA into a smaller fragment of varying lengths. Okay. Okay, the DNA fragment are placed on electrophoresis gel. Okay, so DNA fragment will separate and then to produce the band. When an electric current is applied to the gel, the fragment migrate through the gel with the smaller fragments migrating at the faster rate. After a set time, the fragment bending pattern is ready for analysis and identification. Okay, so now there are the DNA profiling or the bending pattern. Okay, so there we analyze who is the most suspected rapist. Okay. Okay, so the test we will matching the pen with the victims. Okay, so okay, so we compare with the first one. Okay, suspect one. Okay, and then there are the matching. Okay, only 8 matching. Okay, only 8 is not matched. The red one is not matched. Okay. Okay, so we test for the suspect 2. Okay, so a little matching. Okay, so more matching and little not match. Okay, so I know you can guess it. Who is the most suspected? Okay, so based on the evidence, who do you feel most guilty? Okay, so the suspect 2, they has a more matching pattern with the vaginal swab of the victim's blood. Okay. Okay, so the suspect 2 is the most guilty. Okay. So the problem of this criminal case is solved.